Hi, welcome to Microbiology. As a student, I am trying to serve you microbiology in a much simplified and understandable way. Now, for today, I am going to explain the Griffith experiment and Avery, MacLeod, and McCarthy experiment. Now, Griffith experiment. It was done by Frederick Griffith in the year 1928. Okay, and this experiment was done to demonstrate a phenomenon known as genetic transformation or transformation. What is genetic transformation? Genetic transformation is a process of genetic change or alteration. What is alteration? Alteration is change. And what change is done? It is done in a cell. The changes are done in a cell by an exogenic DNA or a DNA which is not from that cell which is transformed. It is from other source or other origin or exogenous origin. Okay, you can see here exogenous origin. Transformation is a one of the three sources of horizontal gene transfer. Horizontal gene transfer means transfer of gene from one bacteria to another. Okay, now one of the types of horizontal gene transfer is transformation, other one is conjugation, third one is transduction. So transformation, we all know that either DNA fragments or plasmid will enter a bacteria. In conjugation, how does it enter the bacteria? It just enters by the help of sex pilus. Okay, by the help of sex pilus. We all know that bacteria has pilus in it or pili. Now through that the DNA enters the other bacteria. The pilus acts as a bridge, between the two bacterias. And in transduction, we all know bacteriophages or viruses. Now viruses are mainly made up only of protein coat and a genetic material. Okay, the bacteriophages just impregnates or it just puts the genetic material inside the bacteria and hijacks its mechanism to produce virions. Okay, now for this experiment, Frederick Griffith in 1928 used Streptococcus pneumoniae, or Diplococcus pneumoniae. Streptococcus is also called as Diplococcus pneumoniae. Okay, why? Because they remain in diploid form. See here, it remains in diploid form 1 2 encapsulated by capsule, a polysaccharide polymer. Okay, polymer of which what things? The capsule is made up of glucose and glucuronic acid. Here, glucose and glucuronic acid. Okay, now the capsule is the most important thing here. Okay, the basis of differentiation between the rough and the smooth colonies produced by the streptococcus, it is the main factor. Now the Diplococcus pneumoniae generally has two types of strains. Okay, one is rough colony producing, one is smooth colony producing. Why does it produce smooth colonies? Just because of the capsule. Okay, the capsulated Diplococcus produces smooth colonies and glistering or shiny colonies due to the presence of polysaccharide. Okay, and the rough colony forming streptococci or the Diplococcus, why they are edgy? They are edgy and rough. They produce rough colonies on the nutrient agar plate. Okay, these are non-capsulated and they are avirulent, and the capsulated ones are virulent. Now for the virulence factor, how does it affect the capsule, how does it affect the virulence? Okay, virulence of the organism. Now the capsule causes immunodisguise. What is immunodisguise? It acts as a cloak or it acts as a protection for the organism. Okay, it acts as a cloak. It hides under the capsule. The Diplococcus hides under the capsule and the immunological system doesn't recognize or doesn't get to know whether an infection has been done by Diplococcus or not. Okay, please remember the monomer units of the polysaccharide which makes the capsule. It is glucose and glucuronic acid. Glucose and glucuronic acid. Okay, now coming to the experiment. For the experiment, let's recap the factors that the Diplococcus produces either smooth or rough colonies. The smooth colony is virulent due to capsule, which acts as an immunodisguise, and the rough colony is non-capsulated and produces rough colonies. Okay, and is avirulent. Okay, the immune system recognizes it due to absence of the capsule and degrades it or destroys it. Okay, now coming to the experiment. When the living R cells, now I have forgot to mention one thing. Yes, the R and S cells, or the rough colony producing and the smooth colony producing, both are having three more types. Okay, the R1, R2 and R3. These are the types of the strain, R1, R2 and R3. And for the smooth one, S1, S2 and S3. Okay, the smooth colony producing organisms have three types, S1, S2 and S3. These are the subtypes. Now, which did Frederick Griffith took for his experiment? He took R2, this R2 for rough colonies, and S3, this S3 for smooth colonies. Okay, he took these subtypes. It is very important to know the subtypes. Okay, it may be asked in MCQs. Now he took the living R cells. What does living means? Which can be grown or that is culturable. Sorry. He took the living R cells, okay, and injected it in a mice, okay, or in a mouse. And what did he see? He see that the mouse lives. He saw that the mouse lives. Okay, the mouse is alive, and he did postmortem. He did postmortem of the mouse and found out that the R cells are present in the healthy mouse. Okay, now why did the mouse live? The mouse lived because the R cells, or the rough colony producing cells. Now for the living cells, living S cells, the smooth colony producing cells. The smooth colony producing cells, when injected in the mouse died. Why? Because died of pneumonia. Why? Because it was unable to, the immune system of the mouse was unable to recognize the cells. Okay, the capsule present in the S cells were acting as immunodisguise or a cloak, and the mouse was unable to recognize the cells, and hence it died. Now when the postmortem was done, there was S cells found from the dead mouse. Okay, now in third part, what did he do? Griffith, he took S cells, heat killed it, okay, such that the cells are no longer alive, and injected, and he injected it inside the mouse, and the mouse lived. Okay, the mouse lived because there was no cells. Okay, there was no cells present in the mouse, and hence it lived. In the fourth installment, what did he do? He took heat killed S cells. Okay, the cells were not alive, and it did not do any harm to the mouse, and along with that he also took R cells, the rough cells. Okay, and if we see normally, it should not have killed the mouse, because both when uh, transferred to the mouse or injected to the mouse individually did not produce any kind of symptoms of pneumonia, and the mouse was alive. But when he injected those two mixed, the mouse died. This was an astonishing fact. Okay, this was a very astonishing fact, that why did the mouse died? Both were non-pathogenic to the mouse when injected individually. So this was the basis of discovery of transformation, okay, or demonstration of transformation. Now this was because there was some factor in the heat killed cells, or the heat killed S cells were having some factors which transformed the R cells. Now when the mouse was done post-mortem and grown on nutrient agar, the R cells were producing smooth colonies. Okay, it was a very astonishing fact that the R cells were producing smooth colonies. Now what was the basis? The basis was that there must be some factors which have transformed the R cells uh, into S cells. Okay, now this is the conclusion part. There must be some factor which caused the change of the R2 cells and made them produce capsule polysaccharide. Okay, and made them produce capsules, and they produce smooth colonies. But he was unable to explain that what factor it was it a protein or a polysaccharide or DNA or RNA? What was it? He was unable to explain. Okay, and this transformation process, then it was not known as transformation, it was known as Griffith effect.